move in and out of the cell, which of course would be via the cell membrane. Now, just four persons actually online, but still gonna continue. Now, in terms of movement, so cells need to move different substances into and out of the cell in order to function properly. So, of course, the different types of cells, they have their different functions. For example, the red blood cells, white blood cells, muscle cells, and the list goes on in terms of the different types of cells which you would have been exposed to while looking at the cell specialization and the different types of cells in the human body. So, in order for those cells to function properly, at times they will need substances such as oxygen and different nutrients, and at another time those cells need to get rid of some substances which the cell itself would have produced, and one such substance would be carbon dioxide. So while your muscle cells, for example, while they are in the process of releasing energy, they not just release energy, they also release a gas that is known as carbon dioxide. Now that carbon dioxide, the cells don't want that carbon dioxide to stay inside the human body for any long period of time. If that is the case, then the carbon dioxide will become what is known as toxic, which of course is not healthy for those cells. So diffusion, osmosis, and active transport are three processes by which this takes place. Now that is, they are referring to the above statement which said that substances need to move in and out of the cell. So if I should pose a question where I'm asking you to give me the different method, the different processes that allow substances to go in and out of cells, then you should give me those three, which of course are diffusion, osmosis, and active transport. So that's the first concept that you need to know in terms of movement of substances that should go in or out of the cell. So we have three ways, which of course we'll be looking at them individually. Alright, so diffusion is the first one we're going to spend some time looking at. As for definition or its meaning, they are saying that it is the movement of molecules from a region where they are in higher concentration to a region where they are in lower concentration. All right. So we have some terms there that we ought to know what we mean by concentration, higher concentration, and lower concentration. Now, in terms of phrasing the definition this way, we could have also rephrased it by saying that it is a movement of molecules from a region where you have a lot to a region where you have a few or small amount. So the terms higher concentration and lower concentration, in layman terms, basically what we mean by higher concentration is from a place or a region where you have a whole heap. You understand, student? Hello?
Yes, sir.
Right. And the
Yes,
place,
sir.
perfect. And the place are
Yes,
the,
sir.
alright, great Latte, great Lewin, I should say. Right, so, and the next term, region of lower concentration, that would be the region where there is small amount or none at all in terms of that substance. Now, looking at the jars below that contain the water and a type of crystal that is known as potassium manganate. Now, if we had face-to-face class, then we would have done this simple experiment where we actually have this type of crystal. Right, so it actually comes with a purplish or a valenda color in terms of its appearance. Now, once you put this potassium manganate in the water, it will gradually change the color of the water from that crystal clear appearance of the water to that purplish look that you're seeing to the extreme right. Now, looking at the glass to the left, then you'll realize that you have the crystal down the bottom there. So that simply means that it is just being placed in the water. Now, if I should ask you to tick or indicate where is a place of high concentration for this potassium manganate, then you should actually show me. You should actually tick the arrow there, because the arrow there is where you actually have a lot of the potassium manganate. So because that is the region or that is the area where you have a lot of it, that is why you would label there as a place of higher concentration, while the rest of the water where there's no potassium manganate, then you would label that as a place of lower concentration in terms of the potassium manganate. Now, moving on to the second, to the glass there in the middle. Now you realize that you actually see some that's some small spots. So where you're seeing those spots, though, that would represent the place of higher concentration. Where at the top of the glass, for example, where there's none, that would be the place of lower concentration. Now when diffusion is taking place, the particles normally collide against each other and actually spread out gradually. So that is why when you look at the glass to the extreme right now, you will realize that the particles, they actually spread right through the entire water. And if you look at it, you can see that it is uniform, meaning that it seems like it actually spread out or distributed equally through the entire water. Now, once diffusion is taking place, where the substances move from a place where there's a whole lot to somewhere where there's none or a small amount, then diffusion will only stop whenever equilibrium reach. The word equilibrium means equality. So equilibrium is actually reached for the last glass, simply because the entire water have the same color right throughout. So that is when diffusion normally stop, when there's what is known as equilibrium. All right, so these boxes now we're gonna use them to further explain diffusion. So the first point is saying, in gases and liquids, these particles move around randomly. So any substance that is moving by the process of diffusion, they normally move randomly. So let's focus on the first box. So we have some particles. We have some that have reddish appearance, and we have some that actually have blue appearance. Now, for this purpose, we just want to focus on the blue ones. Now, you looking at the box to the extreme left. If I should ask you to show me in that box where is a place of high concentration for the blue particles, then you should be labeling the bottom right hand corner, because that's where you actually have all of the blue dots. So that would be the place or the region of higher concentration for the blue particles. Because remember, from the definition, we actually said that diffusion is a movement of molecules or particles from a region where they are in higher concentration to a region where they are in lower concentration. Right. So in looking at the box to the extreme left, the labeling for it, or the notation is saying, at the moment when the blue particles are added to the red particles, they are not mixed at all. So basically you would have the container having the red particles, and then some blue one will be added to it. So the exact spot where the blue one would have entered that container, that's where they are now, because the process of diffusion would have not yet started. Now moving on to the next box, the one to the right of the first one. Now, as you look at it, you will realize that the blue particles now they are starting to spread out throughout the box. So the notation is saying, as the particles move randomly, the blue ones begin to mix with the red ones. And moving on to the third one, as you can see, so the more time goes, is the more they actually spreading out. As the particles move and spread out, they bump into each other. This happens. No, this helps them to keep spreading randomly. So let's say for example you're at home now and you just you pull your ink as in the pen and you have a glass or a cup with some water, because you can do this experiment by using pen ink. Because once you have a crystal, a glass with some crystal clear water, and you should now drop the pen ink in that water, that exact spot where the pen ink actually enter the water, that would be the place of high concentration. But as time goes by, then the water will actually take the appearance or the color of the pen ink, because we have several things that you do at home where diffusion is taking place, just that you would have never looked into it that way. I don't know how many persons know what they call blue. That is what we use to put in the water whenever we are washing white clothes, to make it maintain that white appearance, that crystal clean pretty white appearance. So persons know what we call blue. Lati, Lewin, McFarlane, Powells,
Yes,
Powis. You know what is known as blue? Okay. Alright, so
yes, sir.
blue, you know blue, Lati? That you used to.
No, sir.
You don't know blue that you, when you're washing white clothes, you put it in the water when you're rinsing it.
No, sir.
Alright. So alright, blue, you can get it at the supermarket,
Oh,
or persons who sell like
oh,
bleach
yes,
and so.
sir.
Right. So that can also be used as an experiment, because when you just put the blue in the water, that exact spot where you put the blue, that will be the place of high concentration. Now normally if you just put the blue in the water and start rinse the white clothes, then it will basically spoil the white clothes. What you have to do, you have to ensure that the blue actually mix out properly in the water. Now when you do that, is actually the process of diffusion that is allowing that, because when you mix it out and ensure that the entire ensure that all the blue dissolve, then diffusion actually complete, because all of the water have the same amount, because you would have allowed it to actually mix out. So using blue whenever you're rinsing or washing the white clothes, um, it is a process of diffusion that actually take place, meaning that if you don't allow diffusion to complete when you're using that blue, then the blue gonna spoil the white clothes, because the blue, it is really strong, if you just put it on one spot of the white clothes, then it's gonna actually difficult to get out that blue color from it. So you have to allow diffusion to take place properly there. Whenever you're mixing some juice, like for example you're using Kool-Aid, or you're using tongue, once you throw the water, let's say for example you throw the water in the cup first, and then you put the Kool-Aid and the tongue in it, and you don't stir it around with the fork or the spoon or whatever, then if you start to drink it right away, you will realize that the top part of it will actually fresh. That is simply because the process of diffusion haven't completed. Yes, it would have started, because the Kool-Aid now, once it enter the water, it gonna need time for it to spread out properly. But because we can wait on it, that's why we normally use spoon and fork to stir it. So when we stir it with the spoon or the fork, we actually speeding up the process of diffusion. And you will realize that once you mix that juice properly, the entire juice have the same sweetness in terms of taste. That is simply because diffusion would have reached what is called equilibrium, which means all of the parts in terms of the water have the equal amount or the same amount of Kool-Aid or tongue that you would have been using. Same thing whenever I use sugar. So if I use the sugar to mix some tea or I use the sugar to mix some juice, it is the process of diffusion that allow the entire juice to have the same sweetness once you mix it properly. Because if you just put the sugar in the container, in the cup like that, and pour water on it and start to drink it, then you will realize that the top part of it I got sweet and the bottom part fresh, I should say, and the bottom part of it sweet. That is simply because diffusion still don't complete, so it actually needs some time, or to speed it up then you have to stir it. Alright, so looking at the last box now, where they are saying now the notation is saying eventually the particles are completely mixed and diffusion is complete. Alright, so the last pointer, eventually the particles will spread out evenly, and that is known, what is known as equilibrium is reached. So equilibrium is basically equality. So that simply means that all of the container have the same equal amount in terms of the blue dots. And when you look at it, you realize that the blue dots, they actually evenly distribute throughout the entire box. Alright, so the particles move along a concentration gradient. So once we're talking about diffusion, we have what is known as a concentration gradient. So we now need to understand, we now need to know this concept. What do we mean by concentration gradient? The second point is saying the bigger the difference in concentration between two regions, the steeper the concentration gradient. Alright, so let's look at those boxes down the bottom. So as for A, that is the one to the extreme left. Counting the dots, we have 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15, 16 particles on this side of the box. And the next side where you have it, where the label and say water, you basically have none. So that is 0. So if you have 16 on one side and 0 on the other side, that is a difference of 16. Now looking at the second box now, in terms of how many particles on each side, we have 3, 4, 5, 6, 7, 8, 9, 10, 11, 12 on this side, and we have 4 on the right side. So the difference between 12 and 4 is basically 8. Now the first box have a difference of 16 and the second box have a difference of 8. So the pointer above said that the bigger the difference in concentration between two regions, the steeper the concentration gradient. So if I should now give you these two boxes and ask you which one have the steeper concentration gradient, then you would have to give me the one that have the largest or the greatest in terms of difference, which would be the one to the left. Because if you have 16 on one side and 0 on the other side, that must be a difference of 16. While the second box, you have 12 on one side and 4 on the other side. So that will give you a difference of 8. So the box to the extreme left have a steeper concentration gradient versus the one in the middle, which would have 8. Now in the process of diffusion, the larger or the greater the difference, which would definitely mean that the steeper the concentration gradient. Now diffusion normally take place faster when there is a steep concentration gradient. So again, in looking at the box, so the fact that the first box have a difference of 16 simply mean that if we're comparing the rate at which diffusion going to take place within these two boxes, then the first one, diffusion, will take place faster, simply because that have a steeper concentration gradient. So again, how we determine the steepness of the concentration gradient is basically the difference between the two different regions within that container. Now let's look at the box to the extreme right and see if there's a concentration gradient. So on the left side we have 1, 2, 3, 4, 5, 6, 7, 8 particles on this side. On the other side, when you count it, we also have 8. So and if you realize down the bottom, then say equilibrium, and equilibrium mean equality. So that simply means that throughout the entire region of this box, there is equality, or the particles they are spread out evenly. So the fact that you actually have equilibrium, that simply means that there is no concentration gradient. Because the only time you have concentration exists is whenever there is differences between two different regions. That simply means that one region of, or one part of the container have more of the particles of a particular substance than the other. So the last point is saying that diffusion takes place more quickly when there is a steeper concentration gradient. So if the concentration gradient is not steep, that simply means that there is not much of a difference. Then basically diffusion will take place slow. Alright, so diffusion and cell membrane. So while we're looking at the cell membrane now, it's simply because in animal cell, for example, the cell membrane is what surround the inside, or the cell membrane is what separate the inside particles, or the inside components of the cell from the surrounding. So in terms of substances entering and of course leaving the cell, they actually need to go through the cell membrane, because the cell membrane is the boundary of the cell. So anything that need to leave the cell, or anything that need to enter the cell, they have to cross the cell membrane, and that is what you're looking at. So looking at the screen, the image at the top, where you're seeing that red shape thing that actually look like 11, where you're seeing some substances entering and of course leaving. Now that is what is known as pores within the cell membrane. So the cell membranes normally have some small holes, and these are known as pores. So these pores normally allow small substances to enter or leave the cell based on what instruction is given in terms of what need to enter the cell or what need to leave the cell. And looking at pointer 3 actually give you those small substances, so such as oxygen, carbon dioxide, water, and of course glucose. So these substances, either they're gonna enter the cell or they're gonna leave the cell. So irrespective of which direction they are going, it is the cell membrane that will allow their movement, whether in or out of the cell. So the first point I say, that substances pass into and out of a cell through a cell membrane. So the cell membrane is what is the gateway if something gonna enter or if something gonna leave the cell. Now they continue by saying that the cell membrane is a barrier with tiny pores or holes in it. And I would have just said that we're looking at the diagram now, where we have these two red sections with the gray dots in terms of the particles going in and out of the cell, and those represent the pores. Now large molecules such as starch and protein cannot. So what they are saying is that the cell membrane will only allow some substances to enter. So large substances such as protein and starch, those would be too large. So the cell membrane will not allow those to go through. Now looking at the image below, where you see, so this image represent an organism that is known as amobia. So we can use this diagram to even represent a cell in the human body. Now when you look at it, you're seeing diffusion in and you see diffusion out. Now we have two different colors in terms of dots. We have some blue dots and we have some red ones. The blue represent oxygen and the red represent carbon dioxide. So the arrow, you will realize that diffusion in, you're seeing that it is a blue arrow. That simply means that it is showing the movement of oxygen. So oxygen is basically outside of the cell, and that oxygen need to enter the cell. So the process that will allow the oxygen to enter the cell is what is known as diffusion. Then when you look at the image properly, you will realize that you have more blue dots out of the cell versus the amount that is inside the cell. So because you have more outside the cell, outside of the cell will be the place of high concentration for the oxygen. So that is why it's going to diffuse in. Now looking at the red dots now, which represent carbon dioxide, you will realize that we have more inside the cell than outside. And the red arrow is showing diffusion out. So the carbon dioxide, the fact that more carbon dioxide inside the cell simply mean that diffusion, that process now will allow carbon dioxide to diffuse out of the cell. Because remember that the cell is what would produce the carbon dioxide, you know. And remember I would have said that this carbon dioxide, it need to get out of the human body, because if it don't get out of the human body, then it will become toxic or poisonous. So diffusion is a process that will allow it to basically leave the cell here, for example. Alright, so cell membrane, we normally describe it, we normally call cell membrane a partially permeable membrane, or some textbook will say semi-permeable membrane. And why we classify it as partially or semi-permeable, that is simply because it only allows some substances to pass through. Because remember that looking at the two last pointers, the small molecules such as oxygen, carbon dioxide, water, and glucose, now those can pass through the cell membrane. However, there is a but, because they say large molecules such as starch and protein cannot. So that simply means tell you that only some substances the cell membrane allow to enter or leave. So the word partially and the word semi, those two terms mean some. So we normally said that the cell membrane, it is selectively permeable, or you can say semi-permeable or partially permeable. All of those three terms mean some. Now if I should pose a question and ask you why the cell membrane is described as partially permeable, can you write that question for me, please? It's a question that you will get on test. Why is the cell membrane described as partially permeable? Yeah, you have a question or I should repeat? So the question again, why is the cell membrane described as partially permeable? And the answer for that question is because the cell membrane only allows some substances to enter or leave the cell. Because the cell membrane allows only some substances to enter or leave the cell. Right, so diffusion in the human body. So now we're gonna look at some substances that actually move from place to place in the human body by that process that is known as diffusion. So oxygen is what we will be focusing on now. So the image that you're seeing is a section of the lungs, and that section of the lungs is known as the alveolus, and this is where the human body exchange two gases, carbon dioxide and oxygen. So the human body normally exchange these two gases at some point in your respiratory system. Now, what I mean by that gaseous exchange, are these two gases being exchanged, is simply because when you breathe in, you actually take in oxygen, and when you breathe out, the main reason for breathing out is to give off carbon dioxide. So at some point, these two gases will pass near each other in the human body. So that simply means that the human body is doing a exchange, giving off one to the atmosphere and taking up one from the atmosphere. So the process at which they actually pass each other very closely is known as gaseous exchange. Now that gaseous exchange normally take place in your respiratory system. As to exactly where in the respiratory system, it normally take place in a part of your lungs that is known as the alveolus. So if you're talking about one, you would say alveolus. If you're talking about more than one, then you use the term um, alveoli. So the alveoli is the exact site for the process of gaseous exchange in the human body. So the first point is saying, after breathing in, oxygen diffuses into the small blood vessels of the lungs. Right, and looking at the diagram, so the red this time represent oxygen and the blue represent carbon dioxide. Now when you look at this labeling, so looking at the diagram, you will see where they say O2 diffuses into blood, as O2 mean oxygen. So they're gonna say, let me see if I can get this diagram. I'm gonna get it larger so you can see it. Share screen. Right, so just getting the image larger so you can actually study it. Right, so the yellow, all of the yellow section represent the alveolus in the lungs. Now diffusion is taking place about carbon dioxide and the oxygen. So the red represent oxygen while the blue represent carbon dioxide. So let's follow some arrows. So this arrow that said air move in and out. So this arrow is pointing in. So this simply means that the air is coming from the outside atmosphere into the lungs here. So the air will contain oxygen. Now looking at this red arrow, its labeling is saying that O2 diffuses into blood. So O2 mean oxygen is now diffusing into the blood. So coming that direction. So while the oxygen is going into the blood by diffusion, then carbon dioxide is doing the opposite, where carbon dioxide is leaving the blood by the same process of diffusion, and once it leaves the blood vessel or the blood capillary, it will now enter this part of your respiratory system that is known as the same alveoli, and following this arrow, so the carbon dioxide is now diffusing out of your lungs. So this is what we call gaseous exchange, because gaseous mean they are talking about gases and the two gases they are referring to, of course, they are carbon dioxide and oxygen. So this is where the human body exchange these two gases. Alright, so carbon will diffuse from blood vessel to the air sac in the lungs. So what is known as the air sac is the same alveoli. So when you see that term air sac, they are talking about the same alveoli. So digested food also passes from the small intestine into the blood capillaries by diffusion. So what they are saying is that whenever you consume some food, it is basically gonna leave your digestive system via that process that is known as diffusion. Alright, in terms of the speed or the rate at which diffusion take place, there are some features of those body parts that will be engaging in that process in terms of allowing diffusion to take place simply because of substances moving. So to speed up the rate of the diffusion, there are some things that must exist. Diffusion distance are short. Now what that means is that if there is a substance that going to move from one body part to another by the process of diffusion, they are saying that the closer those two different body parts are to each other, the faster the rate of diffusion will be. Now when you look at the image, you will realize that as I said, the yellow represent the lungs, which are part of your respiratory system, and that section surround that structure that surround the yellow part of the diagram represent a blood vessel, which is a part of your circulatory system. So you will realize that these two parts, even though they are from two different um, system in the human body, they still work close to each other. That means that they're very close. Because if it was a case where, let's say, for example, the yellow part of the diagram on one side of the screen and the reddish or the pinkish part of the diagram, which represent your blood vessel, is on the left side of the screen, then that would be way too far in terms of the distance. So they are saying that the distance between the two different body parts that are going to allow diffusion to take place, they normally stay as close as possible to each other, simply because that speed up the rate at which the substances diffuse. The next thing is concentration gradients are maintained. So in order for diffusion rate to speed up in the body again, there will always be concentration gradient. And remember that concentration gradient simply means that there's a difference between two regions in terms of particles. So in the digestive system, for example, the digestive system itself will always be the place of highest concentration for nutrients, while the blood, for example, or the circulatory system will be a place of lower concentration for nutrients. That is simply because when you eat, when you consume the food, the digestive system is what break down those nutrients. So let's say, for example, some muscles need some protein. Then protein will diffuse from the digestive system into the muscular system by that process that is known as diffusion. So process very important. Now in terms of the speed or the rate, as they are saying, it actually have to do with concentration gradient, which means that there must always be an imbalance. So at any given moment, more energy should be out of the muscles of the cell than what is inside, and simply because of that process of diffusion. So there will always be a concentration gradient once there's a difference, meaning that um, outside of the muscular system, which would be in the digestive system, there's more nutrient present than in the muscular system. So the muscular system will always have to depend on the digestive system to actually get those along with the circulatory system to get those substances move across. Diffusion surface are large. Now what that means is that exact part the exact place in the human body where diffusion is going to take place, there must be large amount, there must be a whole lot of space for this process to take place. Now what you're seeing on the screen now in terms of image, you're actually seeing some alveoli. So those round circular things that you're seeing, which we have 6 of them on the screen, those represent alveoli. So it is inside of these alveoli where the gaseous exchange take place. So if you realize the blood vessels wrap themselves around the alveoli, and that is simply because the oxygen, for example, will diffuse out of the alveoli into the surrounding blood vessel, which will take it to different body parts to give off oxygen, and at the same time pick up some carbon dioxide and take it back to these alveoli so that they can be expelled out of the human body. So ear sacs, when you see that term ear sacs, we're talking about alveoli. So they are saying that the surface area of the ear sacs in the lungs are large because of the millions of ear sacs which are present. So on the screen you're only seeing 6. So they are saying as for real life situation in humans, we have millions of them. So what is taking place in one alveolus is taking place in the other millions of alveoli. So the fact that you have this gaseous exchange taking place in over a million alveoli, that simply means that once you breathe in oxygen, then that oxygen will actually reach the different body parts very quickly because diffusion is taking place at so many different points. Alright, so that should do it for diffusion. So the other process, the other method, the other way by which substances enter and leave cell is known as osmosis. So diffusion basically deal with the movement of particles or molecules, gas. That is diffusion. Now if the cell should need some water or any form of fluid, then osmosis is what should be discussed in terms of that, because osmosis normally deal with the movement of water. Alright, so take a you can take a 1 minute break, student. Just 1 minute, 1 to 2 minutes. Alright, and then I will just continue looking at osmosis. Alright, so osmosis. So they are saying that osmosis is a special case of diffusion. What this means is that the same principles that govern diffusion, where we are talking about movements of substances from a region where it is in higher concentration to lower concentration, is the same thing with osmosis. It is just that there will be a change of terms or explanation in terms of how the substances are moving. Now, osmosis basically involves the movement of water through a cell membrane or through what is known as partially permeable membrane. So it is the same cell membrane that they're talking about, which means that the cell membrane would only allow some substances to pass through, and of course water would be on that list in terms of substances. This means that only some substances can pass through the membrane, and again small molecules such as water can pass through, but larger ones basically cannot. So this pointer is the definition. So they are saying that osmosis is water moving from a region where its concentration is high, so that is moving from a place of higher concentration to where its concentration is low, so from a place of lower concentration. So let's say for example you wash and you have the clothes on the line. I mean, they are not properly dry, but all of a sudden the rain come down. Then when you take up those dry clothes, the fact that not dry clothes, that those half dry clothes are clothes that is not properly dry, then basically the clothes gonna contain some amount of water. Now if you should put those wet clothes or damp clothes on the bed, for example, which is dry, the clothes that is wet would actually be the place of high concentration for the water, while the bed would be the place of low concentration. Now, if you put the wet clothes on the bed and within like 10 minutes or after a few minutes, you go and take up the clothes and feel the bed, then you will realize that the bed is wet. Now, the bed is wet because of osmosis, which means that water will move from a region where it is in high concentration, which would be in the clothes, to a region where its concentration is low, and that low in that case would be the bed. So osmosis is what allow water to move from one place to another. An next example I can use to explain osmosis is, let's say for example some water is spilled on the floor. That means the floor wet up, water there on the floor, and you now should get a piece of dry cloth or a dry mop to mop up the water. Now the place of our region of high concentration for the water will be on the floor, and the place of low will be that dry mop. So water basically gonna move from the place of high concentration to the region where it is lower concentration. So that's how osmosis will allow the water to actually move. So once you dip the mop or place the dry mop in the water, then you realize that the mop gradually start to get wet. That is simply because the water is now moving from a place of the high concentration to the place of the low. Now you will realize that when you're mopping up the water, if there is a lot of water, the mop can only hold so much and no more. That is because the mop basically now have almost the same amount that is on the floor. So equilibrium is basically being reached. So what you'd have to do, you'd have to now wring out the mop and then start to wipe again. So once you wring out the mop, then the mop now becomes a place of low concentration once again. And you continue that process until you basically mop up all the water. So in terms of you mopping up some water from the floor, or if you spill it on the table by using a dry cloth or something that is damp, then diffusion, then osmosis is the process that is allowing that water to be moving. Now when looking at the definition, we said moving from a region where its concentration is high, and in bracket you say a dilute solution. So whenever a solution is dilute, it means that it actually have a lot of water components in comparison um, to molecules in terms of solid molecules. So let's say for example you mix some juice, some sugar and water, whichever juice you mix, and it's extremely fresh, then it would be, that simply mean that it is well diluted. That means that a lot more water particles in it than other substances. Now if you mix the drinks and it is extremely sweet, persons normally say that it is highly concentrated, and it is that sweet or highly concentrated simply because there's not a lot of water in it. So the only way you can get it fresher or less concentrated is by adding water, and we normally say that you're diluting it. So when a liquid or fluid is diluted, when a solution is diluted, it simply means that it having a lot of water. And when a solution is concentrated, it means that it does not have a lot of water. So if you're mixing the juice and it's too sweet, that means that it is too concentrated. That means you don't have enough water. So the only way you can make it get fresher is by, of course, diluting it by adding water, and then know adding the water to it actually make it more dilute rather than concentrated. Now cell in water, so we're now going to look at cells. So on the screen you're seeing plant cell in pure water. So in terms of cell from this plant, if the plant cell is placed in pure water, then osmosis will now allow water to enter the cell, because remember that it is placed in pure water. That means that more water surround the cell than what is inside. So when you place a cell in water, water will enter the cell by osmosis. As if you have one bucket filled with water and you place a plant cell in it, remember it is an entire bucket of water, so obviously more water is surrounding the cell than what is inside. So the cell will become turgid. So once water enters the cell, it basically blow up the cell. So it become what is known as turgid, or if you want to say stiff. So animal cells will swell up and burst if too much water enter the cell. And while looking at diffusion and of course the first part of the movement of substances, we realize that there's a cell membrane. So the cell membrane and the cell wall basically prevent plant cell from bursting. So the plant cell can take a whole lot more water than the animal cell, of course, and that is simply because animal cell is only surrounded by the cell membrane, while plant cell is surrounded by the cell membrane and also what is known as the cell wall. So the strong cell wall stop plant cell from bursting when water enters them. And so in looking at the diagram, so the cell will swell, or time you can say stiff or turgid. So again, the main reason for that is simply because more water is out of the cell, so osmosis will allow water to now enter the cell so that the cell can get the needed amount of water. Alright, so the slide before, of course, that is you placing the cell in dilute solution or water. This time we are looking at placing the cell in concentrated solution. So that means that the cell is placed in something that having, of course, less water than the cell. So water will not have to leave the cell. Now for those persons who eat June plum, when you peel the June plum, for example, and you mix it with salt, put it in a bag. Let's say, for example, you put it in a bag, one of those little small plastic bags. Now, after a while, when you will hold up the bag and look at it, you will realize that there is literally water in the bag. That is simply because the June plum, the cells of the June plum would contain a lot more water than the dry salt that you would have actually placed the June plum in. So osmosis is that process that allow the water to come out of the June plum, because remember that the June plum is surrounded by a concentrated solution, which would be the salt. So it will actually now pull or draw the water from the cells of that June plum until equilibrium is actually reached. So once the cell is placed in concentrated solution, they are saying that water will leave the cell by osmosis. Animal cell will shrink. So if water is leaving the cell in human being, for example, then the cells, they're gonna shrink. So that is why persons are normally asked to consume or drink enough water, because once you're not drinking enough water, then if the blood, for example, or any other body part needs some water, if you're not drinking enough water, then the body actually gonna turn on itself and start to take the water from the cell itself. So they are saying in plant cell the strong cell wall will stay the same. The outside of the cell shrink away from the cell wall. This is called plasmolyze. Now looking at the diagram below, which representing the plant cell again, you will realize that the cell membrane actually going inward. So now normally the cell membrane will actually be within the cell wall, just at the inner part of the cell wall. So as you can see, we just have different aspects of the cell wall where the cell membrane is. I touch on that is simply because it is being shrink. So that process in terms of it shrinking, we normally call it plasmolyze. Alright, so active transport. So active transport is another way by which substances move in and out of the cell. So at first, remember that would have said that we have three different ways. We have osmosis, we have diffusion, and now they are saying active transport. Alright, so students, the first three questions, get out right take down the first three questions. So the first one, name the process by which water enters cell. Right, so from question 1 to question 3. Alright, I'm gonna types another question. Alright, so question 1 to question 6. Alright, so you're gonna complete those questions. Question 1 to 6. Alright, so you're gonna complete those questions, question 1 to 6.